All right, guys. So chapter 18 is about the blood. So the blood is only one component out of three. So if you look at the circulatory system, the circulatory system is comprised of three components. Number one, the blood. That's the first component. One. What's the first component of the circulatory system? It is the blood. Number two, the second component of the circulatory system is the pump that pumps the blood, and that is going to be the heart. The heart is the pump that pumps the blood, and that is component number two. And component number three is going to be, who can guess, is the third component that makes up your circulatory system. Excellent. Those are the blood vessels. Those are the blood vessels. And the blood vessels are either arteries, or veins. So these are the three major components of the circulatory system. Do you know that the heart merges together and comes together with the blood vessels, and they have actually a special system? What do we name that system? Just the heart and the blood vessels. No blood flowing inside them. What do we call those? I'm sorry? Lymphatic system. Lymphatic system deals with a lot of lymph nodes and lymph fluid. There's no blood that is flowing in the lymphatic system or the lymph nodes. It's the cardiovascular system. It's the cardiovascular system. The cardiovascular system is the system that combines the heart, the pump that pumps the blood through the blood vessel. Is that clear? Are we all clear? Okay. The blood itself is the actual fluid that goes through the blood vessels. So today we will begin with this fluid. You know that the blood is comprised of three fluid compartments. Three different fluid compartments. The blood has three different fluid compartments. The first fluid compartment is known as plasma. The first fluid compartment is the blood plasma. The second fluid compartment is the white blood cells. White blood cells. And along with them, we have the platelets. Platelets. We have the white blood cells and we have the platelets. White blood cells, those WBCs, have another name. What's the other name for the white blood cells? Those are the leukocytes. Very good. You have the leukocytes. Leukocytes. And then you have the platelets. Platelets are the band-aids. The band-aids that stick to the injury site and prevent blood loss. Prevent hypovolemia, gunshot wound, laceration where the blood is leaking, the blood is too loose. So those are the platelets. What's another name for the platelets? What's the scientific name for platelets? Thrombocytes. Thrombocytes. We have thrombocytes. You also need to know that this compartment here, the white blood cells plus the platelets. This is called the buffy creamy coat. The buffy creamy coat. That's the buffy creamy coat. What do we find in the plasma? In the plasma we find water, predominantly water, plus electrolytes, sodium and potassium and calcium and magnesium. Okay, and we also find nutrients, the glucose, nutrients such as glucose, lipids, fats, nutrients, glucose, lipids, fats. Are we all following? Nutrients or lipids, fats, proteins, glucose, and we move forward, and we also have toxic waste. Don't think that your blood is only full of useful things. The blood is responsible for a triangle. The blood is responsible for a triangle. Two good things. It brings two good things and it removes one bad thing. It brings in oxygen for aerobic respiration, and we also bring in nutrients and electrolytes, good electrolytes. So we bring in two good things, oxygen for aerobic respiration, and we bring in nutrients. And we remove one bad thing. What is the bad thing that we remove from the tissue? What is the minus? So you know when you bring in this food, when you order food home, do you eat the food and you keep the garbage? Do you just keep it in the house, piling up? One day after another, 1 week after 1 year after. Do you do that? Or do you need something to remove? So you need a sanitation. You need to be able to get rid of the metabolic waste. So same thing. After the tissue metabolizes all of the lipids to get ATP out of them, carbohydrates to get ATP out of them, and glucose to make ATP out of the glucose, then we see that there's a lot of waste that's gonna build up. Same way your car. When you burn the gasoline in your car, you see all the fume coming out, the smoke? That is all harmful waste. So the metabolic waste is gonna be removed. Metabolic, toxic, poisonous waste. Metabolic, toxic, poisonous waste. Is that clear? Is that clear? Any questions? So your blood plasma is where you have the water, the electrolytes, the nutrients, and the toxic metabolic waste that builds up as a result of us breaking down, all of the metabolites. Is that clear? Anybody got a question? Okay. The plasma compartment makes up about 55% of your blood. So if your blood was 1 liter, 550 milliliters is gonna be plasma. That's the biggest part of your blood. If you take a blood sample and you centrifuge it, you centrifuge the blood, you spin it, what's gonna happen to the blood? It's gonna fractionate. The blood is going to lay in 3 layers. 3 layers based on what? Based on color, based on charge, based on what? Based on density, based on how heavy the blood components are. The heaviest will always be at the very bottom. Whenever we do centrifugation, the heaviest element will always be deposited in the bottom. All right, so now the first compartment is plasma. The second compartment is white blood cells, and the third compartment is known as, the second compartment is actually a combination of leukocytes and platelets, and we name that the buffy creamy coat. And that actually makes up less than 1% of your blood. How interesting is that? All the platelets all the white blood cells. What's the function of the white blood cells? White blood cells are there in order to defend you, are there in order to help and defend you from any infectious pathogenic microbe. True or false? Okay, so now if you come into the school, you see 1 or 2 cops. Will you be shocked? If you see 500 cops, will you be shocked now? So that's the same thing with your blood. Your blood usually has white blood cells and platelets in the ratio of 1% because nothing bad is happening. If it was more than 1%, if you have so many white blood cells, it means that there's either infection, that's why they are there. You see a lot of cops, it means there's a lot of problems. So that means there's a pathogenic, infectious, microbial, bacterial, viral, fungal, parasitic infection. Or there's cancer. That's why you have, whenever you have so many cells, that means there's cancer. So always you need to remember those ratios. The blood plasma makes up about 55% of the whole blood, and 1% is actually the leukocytes. And lastly, what do we have? Lastly, we have the RBCs. RBCs are the red blood cells. RBCs are red blood cells. Clear? Question. RBCs are red blood cells. What's the other name for red blood cells? RBC is an abbreviation. Those are the erythrocytes. Excellent. Erythrocytes. Erythrocytes are the red blood cells. How much do you think they make up of your blood? Erythrocytes make up how much? What's remaining? 45%. So 45% of your blood is just red blood cells. And 55% of your blood is blood plasma. And 1% of your blood is the buffy coat, which is a combination of the thrombocytes and the leukocytes. Clear? Anybody has a question? Okay. You need to know that the heart is a pump and it's a pressure pump. The heart is a pressure pump. So blood always has 2 trips. Uptown trip to the lung, that's the lung, and a downtown trip to the tissues. Your tissues, those tissues, they need the tissues. You see, the tissue is made up of a lot of cells. The tissues, and then you have your heart. Your heart has an atria and a ventricle, and then it has another atria and another ventricle, and we will talk about that, but this is your heart. This is the heart, and this is the lung, and this is the tissue compartment. You have 3 compartments. 1, the lung, 2, the heart, and 3, the tissue. And between them is who? The blood vessels. So, blood first is pumped out to the tissues. You see where my arrow is? So, fresh, oxygen-rich blood is sent out. Fresh, oxygen-rich blood is sent out. Fresh, oxygen-rich blood is sent out to the tissue compartment. The tissue compartment is going to take all the oxygen and all the nutrient and burn them like a gasoline. and then it will get all the toxic waste, and the carbon dioxide, and all of that has to be pumped back. Let me put it in blue color because that's our conventional color for deoxygenated blood. That's why when somebody gets punched, there is a bruise, there is a contusion, there is a laceration, you will always see a bluish discoloration of the tissue, meaning there's hypoxia, necrosis, the tissue is dying. So now you see that there is a lot of deoxygenated, carbon dioxide rich and toxic waste. And then this bad blood is going to go back to the heart, and the heart is going to pump the bad blood back to the heart is going to pump the bad blood back into the lungs. This is all bad blood. Bad blood going from the tissue to the heart, and the bad blood going from the heart to the lung, and then the heart is going to replenish this bad blood and make it oxygen rich and nutrient rich. Here, the blood was carbon dioxide rich and toxic waste rich. The blood was rich in carbon dioxide, and the blood was rich in toxic waste. The blood was rich in carbon dioxide, and the blood was rich in the toxic waste. And you see the oxygen and the nutrients are now going to be sent back to the heart. Guys, this is really important. This is really important for you to understand what's happening here. You have three compartments: the heart, the lung, you have the heart and the lung and the tissue. 1, 2, 3. And the three compartments communicate with each other. The three compartments communicate with each other through those arrows represent what? They represent the blood vessels. Now, you see some blood vessels are red, meaning they are rich in oxygen and rich in nutrients, rich in electrolytes, rich in beneficial substance. And you see some arrows are rich in carbon dioxide and toxic waste. Is one better than the other? No, because both have jobs. One job is to deliver you. The Uber Eats person brings you the food, but the sanitation person is also important because they remove the waste from your home. Both are important. You cannot rely on only one. And now you will learn that blood vessels who carry blood away from the heart, we call them arteries. Arteries carry blood away from the heart. And veins carry blood toward the heart. You need to know that. Many students confuse arteries and veins. Arteries always carry oxygen, always carry blood away from the heart, while veins always carry blood where? Towards the heart. Now, students used to say arteries carry oxygen-rich blood and veins carry oxygen-poor blood. Raise your hand if you think that's true. So arteries carry oxygen towards the heart, and veins carry carry bad blood. So in a sense, arteries carry good blood, red blood, and veins carry toxic laden hypoxic blood in them. Is it true or false? Look at the diagram. Is that true? Arteries meaning they are away from the heart, meaning going away from the heart. How many arrows are pointing away from the heart do you see here? Well, this arrow is pointing away from the heart. This one is pointing away from the heart, right? That's the heart. This one is pointing from the heart to the tissue. And what about this one? This one is pointing from the heart to the lung. So you have two arrows pointing away from the heart. So absolutely both of those are what? Arteries. If you point away, if you're carrying blood away from the heart, you are absolutely an artery. So this one here carrying blood from the heart to the tissue, that's the artery. Carrying blood from the heart to the lung, that's the artery. Those are two arteries. But are both of them carrying oxygen-rich blood? Now you see. So arteries and veins are not classified based on what kind of blood is going inside them. Is it oxygen-rich blood? Is it oxygen-poor blood? Is it a blood that is full of nutrient? Is it a blood that has no nutrient? True or false? It's not about the blood flowing inside you. It's about where you're headed. If you're heading to the tissue, or are you heading to the lungs? So here you have arteries with rich, good blood. Here arteries with bad blood. And when it comes to veins, we have two veins too. Veins meaning you're heading to the heart. So that's the first vein heading to the heart. And that's where is the second vein heading to the heart? This one. Do you see them? You see both veins? That's the vein heading to the heart, and that's the vein that's also heading to the heart. True or false? True. Very true. But you see some veins have fresh red blood, and you see veins have toxic blood. So it's not about what the kind of blood flowing in and inside you. It's about where you're headed. If you're heading away from the heart, you are an artery. If you're heading away from the heart, you are an artery. If you're heading towards the heart, you are a vein. So now, how do we name them? If you're heading away from the heart, you either got to be going to the tissues or you're going to be going to the... If you're going to the tissue, we call you systemic artery. Systemic artery. So if I tell you systemic artery, you know you're going to the systemic tissues. So you're not going to be going to the tissue and you got no oxygen on you. You're not going to be going to the tissue and you have no oxygen. You're not going to be going to the tissue and you have no nutrients. If you go to the tissue, you will be... So if I say systemic artery, you already know. I don't need to tell you that it's oxygen-rich blood, that it's nutrient-rich blood, because it's going to the tissue. I'm going to go to the tissue, give tissue bad blood. I'm going to give the tissue good blood. And if I tell you pulmonary meaning I'm going to the lung. Pulmonary arteries are carrying toxic carbon dioxide rich blood to the heart. Is that clear? To the lung. Veins, we have two types of veins too. Some veins are coming back from the lung. We call those pulmonary veins. Pulmonary veins. And you know that the pulmonary veins, they are coming from the lung, so you know they have just been fresh. They just been receiving oxygen, they just been receiving nutrients, and now they are ready to be pumped by the heart. That's why they go into the heart. And you also have systemic veins. That means they are returning from the tissue. Well, if you're returning from the tissue, you're carrying waste with you. The tissue is not going to be giving you good oxygen. The tissue is not the lung. The tissue is going to be giving you metabolic waste to clear, to refresh. So those are, you need to know the moment you hear pulmonary artery, that means we are going to the lung carrying good blood. Pulmonary vein, meaning we're heading to the lung, but it is full of metabolic waste and carbon dioxide. Systemic artery, meaning we are an artery that's going to the system and we are rich in oxygen. Systemic vein, we're leaving the tissue all full of toxic metabolic waste. Any questions? Now, that is we talked about the heart being a pump and it's a pressure pump, and we will talk more in detail about the pressure pump as we proceed, to go to the next chapter. So now, please copy this down and make sure that you submit it. Okay, so I recorded this because I need to do the lecture. So I recorded this, I'm going to post it for you online. You just click on the video and whatever that you need to copy because we need to do the slides. Yes?
Can you just explain again, because I got confused about it, that, you said that pulmonary vein is the one that comes from the lungs over the heart, and it's good.
So vein, you always go into the heart.
Veins always go to the heart.
Veins always heading to the heart. Arteries always leaving the heart. So if you're pulmonary vein, that means what? Well, you go into the heart. There's no doubt about it. You're not a vein. If you're not going to the heart, you're not a vein. You hear the word vein, it means you go into the heart. Now you hear another word, pulmonary vein. What could that mean?
From the lungs to the heart.
Exactly. So if you're going from the lung to the heart, what kind of blood do you think is that? Good, fresh blood, oxygen, rich nutrients. Yeah, there you go. So don't let the word systemic and pulmonary confuse you. Focus on artery or vein. And from there, it's got to be so vein, meaning I'm heading to the heart. Systemic vein, that means I'm coming from the systemic circulation from the tissue. Because I can't be coming from the heart and going to the heart. So the heart is one. You're either going to it or you're either leaving it. So pulmonary vein, I'm going to the heart. Pulmonary, I'm coming from the lung. I'm good blood. Pulmonary artery, I'm going away from the heart and I'm heading to the lungs. So that means it is very nasty, toxic waste blood. Yeah, exactly. All right, so now let's go to the slides are gonna put this in perspective some more. So let us now okay, so now we talked about the circulatory system. You see, and today we're going to talk about chapter 18, and that is the blood. So, blood, from the back in the day, they used to think it gives you power. Some people, even up to this day, people drink blood. People think that if you drink the blood of the lion, it makes you strong like a lion. You drink the blood of the bull, it makes you strong like a bull. No, it will kill you. So you see that blood cells was seen with the microscope, and we see that the circulatory system, we talked about that, the heart, the blood vessels, and the blood, but the cardiovascular system only refers to the, you gotta pay attention to my question and my illustration, because sometimes I put a bonus question. So the main exam questions are those homework that I assigned for you. You want to do Quizlet, you want to screenshot, send it to everybody on the group chat, you can do that. But when you see it on the exam day, you're not going to remember. And also, you carry the lecture, those illustrations I made, you take a photo of them, you post them, and I'm going to make a bonus question, about it. And you're going to be like, oh, I wish I just. So if you just want to do work to get credit, but you're not reviewing it, you got to miss one of the bonus. So you don't need bonus for class. Just the work, the question that I give you in the curriculum. If you do just that, you will do excellent. But if you want to get more points, and I recommend, so just make sure you pay attention to what I do in class and what I discuss. It pays to pay attention. Okay, so the function of the circulatory system, we mentioned two good things and a bad thing. Two good things: to deliver the oxygen, to deliver the nutrients, and to remove the metabolic waste. Don't underestimate that. People who always get heart attack, it's because oxygen is not arriving to the heart tissue, muscle, to the heart muscle. It's because nutrients are not arriving to the heart muscle, and because metabolic waste is building, sedimenting, depositing, and then the heart is going to be suffocating and dying. So the blood is a transport system that is responsible for, so we're here from 1 to 3, and from 3:30 to hold on. So we're here from 1 to, I'm sorry?
1 to 4, 4:30 to 6:30.
But we can cut that.
No. We want as much knowledge, as wealth of information as possible. So we're here from 1 to 3:30, and from
1 to
4
4,
to
1 to 4:30, 6.
oh, that's a good thing. 1 to 4:30 is good.
4:30 from 4:30 to 6:30.
We have a 30.
1 to 4, and then from, that sounds better. 1 to 4, and then from 4:30 to 6:30. You get a break. Okay, so we transport the oxygen for aerobic respiration, and we remove carbon dioxide and metabolic waste. We remove carbon dioxide and metabolic waste from building up and toxic poisonous waste. Okay, so inflammation is going to require the white blood cells to be transported by the blood. So we talked about the white blood cells, those leukocytes, the neutrophil, the eosinophil, the basophil, all of those cells. They need to be transported by the blood to this tissue that is suffering from the infection. Let's say some bacterial infection on my skin, bacterial infection on my lung, I have pneumonia, bacterial infection in my intestine. Those are 3 different places. Blood is going to be carrying the white blood cells wherever the infection is, and that is called inflammation. You guys need to remember that. Inflammation from flame. Inflammation, inflammatory diseases are always associated with redness, heat, pain, swelling, and loss of function. Redness, heat, pain, swelling, and loss of function. Any questions? Okay, so blood is important for, you remember, for transporting oxygen, carbon dioxide, nutrients, waste, for protection, meaning that your blood is the one who is bringing the cops, bringing the Air Force, bringing the Navy, bringing all of the neutrophils and the basophils and the eosinophils and the lymphocytes in order to fight and kill all the infectious microbes. The blood is also important for regulation. You know, inside the body, you have the plasma compartment full of water, and then you have an interstitial compartment, and that is the compartment that is zigzag between the tissue compartment and the blood. So you don't only have blood and tissue. When I drew that diagram, it looked like you only have blood, tissue, lung. No. Between the blood and the tissues, there is a third compartment known as the interstitial compartment. Is a middleman, is a mediator between the, that's how people get edema. What is edema? Edema is when water is sequestered in the interstitial compartment of the tissue. So we will talk more about that. Regulation, meaning that we stabilize the pH and the temperature. You know that the blood is rich in plasma, full of water, so the sweat. How does your sweat gland make sweat? Because they take water from the plasma. They take water from the plasma. Cerebrospinal fluid. Anybody heard of cerebrospinal fluid? Where do we get that from? From the blood plasma. The saliva. Where we make the saliva from? From the blood plasma. So the water in the plasma, the body uses it for many important vital functions like salivary gland, lacrimation, tears, the sweat glands, the cerebrospinal fluid, the fluid in your joint, the synovial fluid in your knees, that's also from the blood plasma, the water in the blood plasma. Okay, we have about 4 to 6 liters of blood, and the blood is, you guys talked last semester about the different types of tissues. What are the different types of tissues that you guys covered last semester? There were 4 types of tissues. Epithelial tissue in your skin, and muscular tissue, and nervous tissue, and the last one is? Blood tissue is not a category. It's the connective tissue. So blood is a type of connective tissue. Like the bone is a type of connective tissue. The blood is a type of connective tissue. The bone is a type of connective tissue. And you also have cartilage. Cartilage is an example of connective tissue. There's many different types of tissues. So the blood plasma is, if you look at the blood, if you take blood plasma, I mentioned to you earlier that blood plasma is rich in water. But does that mean it's all completely water? That doesn't make any sense. Blood plasma has water, but what else does blood plasma have in addition to water that I already mentioned? Electrolytes, excellent. What else? Just water and electrolytes? Nutrients such as sugars, carbohydrates, lipids, fats, proteins, and also amino acids, electrolytes, vitamins, neurotransmitters, hormones. Talked about hormones last week. They flow in the blood plasma. So in blood plasma you have formed elements, and the formed elements represent the blood cells and the cell fragments. So you have to understand that platelets, remember what I call them? Did I call them cells? Did I say platelets are cells? No, platelets are band-aids. They are fragments of cells. Platelets are chopped off, break off. You know how you break bread? That's plasma. Platelets break off cells known as megakaryocytes. Megakaryocytes. If I write on this thing, it will mess up, right? Are called those are called megakaryocytes. Those are megakaryocytes. Any questions? Any questions? Everybody knows megakaryocytes? That's the big cell from which we break the platelets. You're right, thrombocytes. And white blood cells, the leukocyte. You know if you end with site, what does site mean? Site means cell. Thrombocyte, platelet, leukocyte, white blood cell, and red blood cell is erythrocyte. So if you are a cell, you are a site, you're called formed element. You are formed. So the plasma is rich in formed elements, red blood cells, white blood cells, platelets. I know some questions on the homework, which of the following is not a formed element? By the way, some other books, they don't call them formed elements, they call them preformed elements. Preformed elements. Same thing, tomato, tomato. If you hear the word preformed, what? The professor said formed. Same thing. What? It's the same thing. It's a good narration. Okay. Now let's look at the the three types of the formed element. We talked about the red blood cell. Please remember something. I say this to all my students. Please remember that erythrocytes are red blood cells and they are discoid, looking like a disc, discoid, looking like a disc. Not a donut. Most of the students say red blood cell look like a donut. No, the donut has a hole inside it. Red blood cell has no hole inside it. And you know that the red blood cell is biconcave, meaning is doubly depressed, from both sides. It's called biconcave, meaning it's depressed from both sides. Biconcave, and discoid. The reason why they are biconcave and discoid, there's got to be a reason for that. What's the reason why God made the red blood cell biconcave? No reason. It's just a coincidence like that. No, there's a reason. Because when the red blood cell is biconcave, it maneuvers, flipping all over like a frisbee. You know when you spin the frisbee, it maneuvers really well in the air. Red blood cell is biconcave because it maneuvers really well where? In blood vessel. Not in the blood, it is the blood. It's in the blood vessel. In the blood vessel. In fact, the spleen, you know the spleen? Where is the spleen found? Right side or left side? On the left side. The spleen is known as a nickname. What's the nickname of the spleen? I heard it. Very good. Is the graveyard of red blood cell, because the spleen is full of sinusoids. Sinusoids are blood-filled cavities. You know the show American Ninja Warriors, where you have to jump and do some tricks and make it through. Those really young folks, they can do it. Me, I can't do it. So the same thing, that's what the spleen is there for, to do that for the red blood cell. Are you a young red blood cell? We'll get in there, let's find out. So the red blood cell, if you're young and fresh, it'll make it through. If it can't, it'll fall and get trapped in those sinusoids. And there are macrophages, killers, shredder, like the shredder, to kill you if you fall in there. They are called sinus histocytes. It's good to be young and light. It's not good to be old and sluggish and big. Today in a graduation, they called the name of this young graduate. She kept running. I'm like, oh, I wish I could do that. I cannot do that. She came like lightning fast. She got on the stage, picked up her award, spin, and I'm dizzy just looking at that. She was just so fast. So red blood cells, you know how old the red blood cell can last? 3 months. That's why you do the A1C test. You do the A1C test every 3 months. Because when a red blood cell is just fresh born, where is the red blood cell born? Red bone marrow, in the bone marrow. So they just come out, they are just like that student, very fast. She's gonna be having fun working as a nurse. And then those really big, heavy, very heavy red blood cell, it will be very hard for them to make it through the American Ninja Warrior test. Anyways, so the erythrocytes undergo this challenge of durability in the spleen. Okay, platelets. Remember, I like this word, cell fragments. Then why do you call them thrombocytes? Just to confuse us. but they are not cell. You know now everybody is a cell. The sperm is a cell. Even though they have half chromosome, they're still a cell. The egg is a cell. They got half chromosome, is a cell. The bacteria got no nucleus, still a cell. Everybody's a cell now. So now the thrombocyte is a cell too. But it's not a cell. It's a fragment. The leukocytes, there are 5 leukocyte types divided into 2 categories. The granulocytes, we call them the BEN, B-E-N. Basophils and eosinophils and neutrophils. So basophils stain basic, bluish, blue basophils basic, BBB, blue basic basophils. Eosinophils are red, acidic, they stain acidic. And neutrophils, they're neutral, they're staining with no color. But one of the big high yield concept is that neutrophils always target who? Neutrophils always target the bacteria. And basophils, they target the viruses. And eosinophils, they target the parasites. A granulocyte, you really need to know that. What's a granulocyte? Granule means a bag. So when I show you a photo of the basophil, neutrophil, and eosinophil, they are full of bags. Why are they full of bags? Why is the neutrophil, the basophil, and the eosinophil full of bags? Because what? They're full of bags because they're full of bags because the bags are full of weapon to kill the bacteria, right? Acid, the bag is full of acid to burn the bacteria. Acid, degradative enzymes, proteolytic enzymes, metalloproteinases, those are all weapons that destroy the invading pathogenic bacteria. Eosinophils, neutrophils, and then agranulocytes. Agranulocytes have no granules, and those are the lymphocytes and the monocytes. Those are the lymphocytes and the monocytes. Those are the detectives. You know, first, the granulocytes are generalized. When you call the cops, the same cop will show up for check fraud. The same cop will show up for robbery. The same cop will show up for assault. The same cop will show up for homicide. But when a problem gets bigger, now you get a specialized robbery detective, homicide detective, check fraud detective. You get it. You get it, guys?
Yes.
But the lymphocytes are the detective. Monocytes, what are monocytes? Monocytes are huge lymphocytes. Monocytes are huge agranulocytes without granules, and they circulate immaturely. They circulate immaturely. They're immature. They circulate immaturely. And once they circulate immaturely, they're immature, but whenever they find an infection, they differentiate. Remember the Pokemon? Everybody knows Pokemon? So Pokemons, they say they used to what? Evolve. You say they evolve. You see, it's interesting. I watched it in another language. But yeah, they call it evolve. That's exactly what happens to the monocyte. So the monocyte is a big old monocyte. When it finds something dangerous, it will evolve, but we don't say evolve here. We say differentiate. We use the word differentiate. Differentiate or proliferate. Proliferation, differentiation, proliferation, differentiation. So those monocytes are going to proliferate and differentiate. There we are. Now look at a neutrophil. You see the granules? See the granules, the dotted granules? Basophil. You see the granules, the bags, the balloons? Do you see the neutrophil with granules? Now, when you look at a monocyte and a lymphocyte, do you see granules? They are agranulocytes. Very important to know that. Now, we talked about that. Now, when the blood plasma, when you take a blood sample, you put it in a centrifuge, spin it, and when you spin it, it's gonna break apart into 3 layers. We talked about those. The topmost layer is about how much? 55% of the whole blood. That's actually the complex mixture of water, proteins, nutrients, electrolytes, nitrogenous waste, hormones, gases. That's the 55%, but it's the lightest. You need to know them. You need to know the 3 names. You need to know the structure. You need to know who's at the top, meaning the lightest, who's at the bottom, meaning the heaviest. In the middle, you have the Buffy coat. That's the leukocyte and platelets, where they make up about less than 1%, which is good. We want it that way. We don't want our white blood cell becoming 10%. That means something crazy happening, or cancer, God forbid. And we have the erythrocyte, about 45% of the whole blood. Plasma is a liquid portion of the blood, and the remaining is the remaining fluid when the blood clots and the solids are removed. It's identical to plasma. However, you need to know the difference between serum and plasma. You will see that serum is identical to plasma. However, serum is missing one thing. What is it? Fibrinogen. Serum has no fibrinogen. There are 3 major categories of plasma proteins: albumins, globulins, and fibrinogen. Those are the plasma proteins you really need to know. Albumin is the one that keeps the osmotic balance to prevent water from leaking into the tissue compartment and causing edema. Most of the people that their blood, so people who have kidney failure and the proteins are being lost in the urine, that disrupts the osmotic balance, so water leaves the blood and goes to the tissue to make tissue edema. So the albumins, they are the smallest, but they are the most abundant. They contribute to the viscosity and the osmolarity. We just talked about that. They lock the water in the intravascular compartment, preventing it from leaking out to the extravascular compartment in the interstitial fluid and the tissue compartment. There are globulins. Globulins are the main structures on which we make the antibodies. So they provide immune system functions and are important for immune system function. And fibrinogen is a precursor of fibrin threads that help form blood clots. Precursor of fibrin threads that help form blood clots. And you see the name here, nogen, fibrinogen, precursor of fibrin threads that help form the blood clots. Clear? Precursor of fibrin threads that help form the blood clots. Plasma proteins, they are formed by the liver, except globulins, those are produced by the plasma cells. They are produced by the plasma cell. Nitrogenous compounds, those are the waste. So that's why people who have liver disease, they have a problem making the blood plasma proteins that are important. Remember, the blood plasma proteins are fibrinogen. Fibrinogen is important for what? I'm sorry? To form clots. Make the blood more viscous to prevent blood loss. Very good, to prevent blood loss. Nitrogenous waste. You have urea and its toxic end products of catabolism, and they are normally removed by the kidneys. So the waste is always being removed. You see, this is the toxic end product of catabolism. Nutrients are glucose, vitamins, fats, cholesterol, phospholipids, and minerals. Dissolved oxygen, carbon dioxide, and nitrogen is in the blood plasma. And electrolytes, you see sodium makes up about 90% of plasma cation. Viscosity. Viscosity is the resistance of a fluid to the blood flow, resulting from a cohesion of its particles. So whole blood is 4.5 to 5.5 times as viscous. Viscous means thick. So honey is more viscous than water. Ketchup is more viscous than water. What is more viscous, ketchup or honey? Honey. Honey is more viscous. And plasma is 2 times as viscous as water, because obviously plasma is not only water. It has water and it has a lot of electrolytes and proteins. The osmolarity of the blood, that's the total osmolarity of those dissolved particles that cannot pass through the blood vessel wall. So if too high, the blood is going to absorb too much water, that's going to increase the blood flow. If too low, too much water stays in the tissue and the blood pressure drops, then edema occurs. So the optimal osmolarity is known as the U osmolarity. It's called as the U osmolarity. We don't want hyperosmolar blood, and we do not want hypoosmolar blood. So when you're running the marathon and you're so dehydrated, what happens to the osmolarity of your blood? How does the blood overall osmolarity? The blood osmolarity is usually 300 milliosmoles. If you run the marathon, lose water, sweat for 2 hours. Do you think the osmolarity is going to remain at 300 milliosmoles? What is the osmolarity going to become? More than 300 or less than 300? Less. No, it will become more. Osmolar means full of solutes. So before, the water was dissolving the solutes. Now the water has been evaporated in the sweat. So now the solutes are more. So the blood now is much more osmolar than before. So when the blood is hyperosmolar, what happens to the red blood cell? It's going to shrink, because all the water inside the red blood cell is going to be leaking out from the inside of the red blood cell to the plasma. And then what happens to the red blood is going to shrink. And once you get to the finish line and you drink more water, so much water, now there's so much extra water in your plasma, your blood is going to become what? Hyper or hypoosmolar? Hypoosmolar. And then the water is going to start leaking into the inside of the red blood cell. Then the inside of the red blood cell is going to become swollen. This is hypoproteinemia, and that's one of the biggest diseases that is caused by hypoproteinemia, known as Kwashiorkor's disease. Kwashiorkor's disease. Now, if you look at hypoproteinemia, it means from the name. The name is very revealing and telling. What is hypoproteinemia? Hypoproteinemia means that the blood is suffering from a sharp, severe drop in what? Sharp, severe drop in sharp severe drop in hypoproteinemia. Sharp severe drop in protein level. Sharp severe drop in protein level. Now, what is that going to do to the blood osmolarity? If you had a blood plasma that was rich in proteins, now the proteins are gone. What's going to happen to the overall osmolarity? Is it going to stay at 300 milliosmoles, or is it going to increase, or is it going to drop? It's going to drop. So when the osmolarity drops in the blood, water is going to leave the blood and it's going to go where? Who's near the blood? Remember, you have two compartments, the blood compartment and the tissue compartment. The blood compartment and the tissue compartment. When the blood compartment protein drops, the water will say, I don't have anything to dissolve here. Water is a versatile solvent, dissolves. Then water is going to flow from the blood into the tissue. From the blood into the tissue. That's why you see they have big bellies. Because they are starved. They are starved. These little babies are starved. There's no protein in the food that they eat. It's malnutrition food. So the blood is lacking protein. So the water leaves the blood, goes to the tissue of the abdomen. That's why they have these big bellies. Clear? What is sort of mind of that picture is funny? Do you think so? Yeah. One student was laughing at this photo. In the other class. Interesting. It was in this actually same thing. I taught this course many years. This is the second time I teach in this room. The student who laughed at these children was in this seat actually. What is funny? Yeah. Kwashiorkor is funny. If he gets it, he will not be funny. It's funny when it happens to other people. Okay, so children with severe protein deficiency. You need to know the rationale behind Kwashiorkor disease to be able to explain why do these little children have big bellies. It's because of the waters flowing into the tissue. Now, you need to know how the blood is being made. Blood is made in the, where? Bone marrow. So somebody who smokes smoking a cigarette, that person is in the biggest trouble in the world. Cigarette, vape, weed, hookah, all lead to hell. Because when that person is now smoking the nicotine, disrupting the nicotinic receptors in the ganglia, what happens to the blood? What is the effect of nicotine on the blood, on the red blood cell? Some students said it destroys it. It doesn't destroy it. It doesn't damage it. If you spray nicotine on the red blood cell, it will not destroy it. But it will prevent it from doing what? From unloading oxygen. So you guys are so many cells, and together you make a tissue. I'm a red blood cell. My job is to bring oxygen and deliver it to you, the tissue. If the person smokes cigarette, nicotine, the nicotine is going to prevent the red blood cell from unloading oxygen. Well, what is the bone marrow going to do? The student said it will make oxygen. Is the red bone marrow a tank? Is it an oxygen tank? It can never make oxygen. The bone marrow will never make oxygen. But it can make more and more red blood cells in hopes to accommodate more oxygen in the blood. So if you go up in the mountain, there's not enough oxygen. If you go up in altitude, is there more oxygen or less oxygen? There's less oxygen. There's more oxygen that down here. So in the airplane, there's an oxygen pump, pumping air inside the airplane. True or false? When you walk up in the mountain, there's not enough oxygen. Now you take a deep breath, your bone marrow is inspecting, tasting your blood. It doesn't have enough oxygen on it. So it will make more red blood cell so it can absorb more oxygen. Well, how could that be a problem? Why is that a problem? Why is making too much red blood cell? I thought the more the better. The more red blood cell, the better. Why is it when you make a lot of red blood cells, the oxygen is a big problem when you make a lot of red blood cells. When you make a lot of red blood cells, the blood becomes ketchup. The blood becomes very viscous. Right or wrong? You're making more red blood cell, more red blood cell, more red blood cell. Hey, there's too much red blood cell now. So why is that dangerous when the blood becomes too viscous, too thick? Blood clots. Make blood clog the blood vessels. You die. Clear? Questions? Production of the blood vessels, especially the formed elements. Hematopoietic tissues produce blood cells. Yolk sac produces stem cells for the first blood cell, and they colonize the fetal bone marrow, the liver, the spleen, and the thymus. The liver stops producing the blood cells at birth, and the spleen remains involved with the lymphocyte production. The red bone marrow produces all formed elements. Remember, whether it's a basophil, or a eosinophil, or an erythrocyte, no matter what it is, always the red bone marrow is the one who's creating. There are pluripotent stem cells. Those are formally called hemocystoblasts. There are colony-stimulating units or colony-forming units. Those are specialized stem cells only producing one class of formed element. Myeloid hematopoiesis, that's blood formation in the bone marrow, and lymphoid hematopoiesis is blood formation in the lymphatic organs. So now let us go into the erythrocyte. So I want to ask you a question. You know, we talk about stem cells. What are stem cells? Cells that make more cells. Who want to add to that? Cells that make more cells. What are stem cells? So stem cell, you know how you make dough. You know dough? Flour, water, eggs, butter. You mix them all together. Then that's your raw material. You can take the dough, make cake. Take the dough, make pancake. Take the dough, make brownies. Take the dough and make bread. So the dough is the starting material for all these different products, right? Stem cell is the original cell. It's called multipotent. It has multi-potential. It can be a red blood cell. It could be a white blood cell. It could be erythrocyte. It could be a platelet. It could be basophil, neutrophil, eosinophil, lymphocyte, monocyte. So the same starting point, but it will, remember how we said the Pokemons evolve? But this one here, you know, each Pokemon has to become one thing. But this stem cell, it can become any one of them. So we don't say the word evolve. What are the two words that I told you today? Differentiation, and proliferation. What do we call it when you look at a red blood cell like that? That red blood cell is concave. So you have to understand that red blood cells have two jobs. If you ask a student, what does a red blood cell do? The student will say it will transport oxygen. Red blood cell will transport oxygen, and they will forget that it will transport carbon dioxide too. Because if you leave behind your carbon dioxide in the tissue is going to suffocate and die. Insufficient red blood cells may kill in minutes due to the lack of oxygen to the tissue. There we go. That's a discoid red blood cell, and that's a biconcave red blood cell. So the disc-shaped cell with thick rim it is loose, and nearly all the cells during the development, they lack mitochondria, anaerobic fermentation, and the lack of the nucleus and the DNA. So there is no protein synthesis or mitosis. Form and function. The blood type is determined by the surface glycoprotein and the surface glycolipids. The cytoskeletal proteins like spectrin and actin, they give the membrane durability and resilience. Form and function. So we talked about red blood cells. They are important for transporting the oxygen and removing the carbon dioxide. For transporting the oxygen and for removing the carbon dioxide. So there is an increased surface area volume ratio that allows for the accommodation of oxygen and delivering it to the tissue. So the major function of red blood cells is increased surface area over the volume ratio, and due to the loss of the organelles during the maturation, there's an increase in the diffusion of substances. 33% of the cytoplasm of a red blood cell is hemoglobin. And oxygen delivery is to the tissue, and we know that carbon dioxide is sent to the lungs. Now I want to introduce a very, very crucial, very important enzyme known as Ca. What is Ca? Ca is carbonic anhydrase. So carbonic anhydrase is found in the cytoplasm of the red blood cell. Produces carbonic anhydrase from carbon dioxide and water. So that's the enzyme. Carbonic anhydrase converts carbon dioxide and water to make for us carbonic acid. Carbonic acid is H2CO3. Its important role is in the gas transport and balancing of the pH. Now let's look at the structure of hemoglobin, which is very important. Hemoglobin is a quaternary, meaning 4 subunits. It's a quaternary structure. It has 4 subunits. Now each hemoglobin molecule consists of 4 protein chains or globins. 2 alpha and 2 beta. 2 alpha in purple and 2 beta. 2 alpha proteins and 2 beta proteins. True or false? True or false? 2 alpha proteins and 2 beta proteins. Non-protein moiety. So the heme group now, so you have to understand, there is a heme group, there is a red seat, a heme group on each proteinaceous subunit. So how many alphas and how many beta? 2 alpha, 2 beta. Each alpha has one heme group, and each beta has one heme group. On the heme group is iron, and the iron binds actually the oxygen. So non-protein moiety that binds the oxygen to ferrous ion at its center. So remember, there is ferric meaning Fe plus 3, severely oxidized, lost 3 electrons, Fe plus 3, ferric. Ferrous is what we love, Fe plus 2. That's the form, the ferrous iron is the form of iron that will bind oxygen on the heme group on each of the 4 subunits. So that is hemoglobin. Fetal hemoglobin, babies have something known as fetal hemoglobin. The only difference is structurally, fetal hemoglobin is, it has 2 alpha, fetal hemoglobin has 2 alpha subunits and 2, no beta, gamma, 2 gamma, 2 alpha subunits and 2 gamma subunits. Questions? Okay. So now, the non-protein moiety that binds oxygen is called the heme group to the ferrous ion at its center. Globins, 4 protein chains, 2 alpha and 2 beta chains, that is 5% of the carbon dioxide in the blood is bound to the globin moiety. Red blood cell count and hemoglobin concentration indicate the amount of oxygen the blood can carry. So now we need to know what is hematocrit. Hematocrit is the cap cell volume, which is the percentage of the whole blood volume composed of red blood cells. So the ratio of the volume of red blood cells to the total blood volume, that is the ratio of the hematocrit. So how much red blood cell do I have in my blood? Remember that 45% that we drew from the centrifuge when we spin the blood? 55% was the top plasma, 1% was the buffy creamy coat in the middle, and 45% in the bottom. That's the hematocrit. Just purely red blood cells, nothing else. That's hematocrit. So values of hematocrit are lower in women because androgens stimulate red blood cell production, and women have also the menstrual cycle. You see a lot of these young females in the menstruation age, and they lose a lot of blood in the menstrual cycle. So they need to take the iron pill in order to, hemoglobin is rich in ferrous iron. So we need iron in order for a bone marrow to take it and make hemoglobin for us. But if you have anemia, that is iron deficiency anemia. You have the anemia that is caused by a deficiency in the iron concentration. Questions? Any questions? That's what I was talking about in the beginning. That this is a pluripotent stem cell. Then it's going to become the moment you become a colony-forming unit, you can never go back. So there's a colony-forming unit for erythrocyte, so we call it colony-forming unit, erythrocyte colony-forming unit. There is a basophil colony-forming unit. There is a eosinophil colony-forming unit. There is a neutrophil colony-forming unit. But once you go from a pluripotent, multipotent stem cell into a colony-forming unit, that's it, you're going there. So think of it this way. All of you and your friends in the high school, you were all high school students. But once you go into college, you have committed to a track. You're either going to become a nurse, you're either going to become a pilot, you're either going to become a chef, you're either going to become a medical doctor, you're going to become a police officer. So although you used to have the same potential, you had the potential of becoming whatever you wanted. But once you enter the college, you committed to one track. So the colony forming unit is the very first commitment in the successive stages that give you the ultimate desired preformed or formed element. So pluripotent stem cells give you erythrocyte colony forming unit. Then the colony forming unit for erythrocyte will give you erythroblast. The erythroblast will give you a reticulocyte. Then the reticulocyte will give you erythrocyte. 2.5 million red blood cells are produced per second, and an average lifespan of 120 days. 120 days divided by 30 is how much? 4. So that's why every 4 months we're going to do. So the age for the red blood cell is we do it every 4 months. Every 4 months, a new red blood cell is coming out. So we do 3 times. We do 3 times a year. Okay? Now, first committed cell we talked about, that's the erythrocyte colony forming unit. And the nucleus is discarded to form a reticulocyte. You need to understand that the reticulocyte is going to be the last step that will give us the mature cell. You see, this is very important. This is the iron metabolism. So we need to be able to have a system that recycles. A system that recycles for us iron. It's a mixture of ferrous and ferric. When you ingest iron, it doesn't come in the form of Fe+2, which is the desired form. What is the name of that iron? Fe+2. What do we call that? Fe+2. What did we call Fe+2? Iron that has been oxidized twice. Fe+2. What do we call that? Ferrous. Ferrous iron. But Fe+3 is known as what? Ferric. So now they're both mixed. There's a mixture of ferrous and ferric. The stomach acid, the burning acid that is released by the P cells of by the chief cells of your stomach is going to convert ferric into ferrous. That's what we need. And then gastroferritin is going to be the submarine that will transport for us ferrous. Not ferric, ferrous. Then gastroferritin is going to transport ferrous to the small intestine and releases it for absorption. In the blood plasma, ferrous is going to bind to transferrin. So gastroferritin, and you can get a hint from the name gastro, meaning that is only in the stomach. So it's going to transport ferrous to the small intestine and is going to dump it in there. So gastroferritin is only working in the gastric mucosa of your stomach. Then transferrin will take the next step. It will take iron and it will transport it in the blood plasma. Ferrous will now leave gastroferritin and will now bind to transferrin. And transferrin is going to go to the liver. So in the liver, some transferrin is released. Is going to release ferrous for storage. And then ferrous that has been released for storage in the liver is going to bind apoferritin to be stored as ferritin. So ferritin is actually a stored element of ferrous that has been brought to the liver by the transferrin. So transferrin hands over ferrous to apoferritin, and apoferritin transports it stored in the liver as ferritin, and then the remaining transferrin is going to be distributed to other organs where ferrous is used to make hemoglobin and myoglobin. That shows you how important ferrous is. So let's look at the different important roles that ferrous has. You eating food, it has a little bit of ferrous, a little bit of ferric. No problem. If you're ferric, who's going to convert ferric into ferrous in the stomach? Acid. Excellent. So now we all have ferrous. Who's going to be transporting ferrous inside the stomach, gastric, acidic mucosa, and handing the ferrous to the intestine, away from the stomach into the intestine? What's the name of the protein that does that? Gastroferritin. It's going to transport ferrous to the small intestine and release it for absorption. Once you're in the small intestine, you're not yet done, because ferrous is going to be passed to transferrin, and transferrin will go to the liver and pass transferrin, the ferrous, into apoferritin, and apoferritin will store it as ferritin. So the remaining transferrin, remember the job of transferrin is to release ferrous for storage in the liver. So transferrin gives ferrous to apoferritin in the liver, and apoferritin stores ferrous as ferritin. The rest of the ferrous that has bound to transferrin is going to be distributed to the other organs to make hemoglobin and myoglobin. We talked about iron being the key nutritional component, and we talked about the ferrous and the liver. Okay, you need to know that vitamin B12 and folic acid, so rapid cell division and DNA synthesis occurs in the erythropoiesis. Vitamin C and copper are important cofactors for enzymes synthesized in hemoglobin, and copper is transported in the blood by an alpha globulin called ceruloplasmin. Now, this is what I was talking about earlier. This is what I was discussing with you guys earlier. That whenever oxygen drops, there are oxygen sensors. They taste the blood. Remember, I told you the blood is being tasted constantly to see if the sugar is too high, sugar is too low, if oxygen too high, oxygen is too low, that kind of thing. So the first step is, if there is hypoxemia, hypoxemia, does that mean there is too much oxygen or too little oxygen? Hypoxemia means that there is too little oxygen. The too little oxygen is going to be detected by the liver and the kidneys, because blood goes to the liver and blood goes to the kidneys. So they're going to sense the oxygen. They're going to secrete erythropoietin. You really need to know the name of this enzyme. Erythropoietin. EPO. What is erythropoietin? What is erythropoietin? It's a hormone enzyme that is secreted by the kidney and the liver, and it will, once it goes and binds to EPO receptors on the bone marrow, what will the bone marrow do? It will make more red blood cells. So that's what I was talking about earlier. When you're smoking nicotine, and nicotine is going to do what to the red blood cell? What is nicotine going to do to the red blood cell? to the red blood cell, it's going to prevent it from unloading oxygen. So the red bone marrow is going to think that there is no red blood cells around. It has no way of knowing, no, we do have enough red blood cell. It's the nicotine preventing them from releasing the oxygen. Okay? So this is stimuli for increasing erythropoiesis, which is low levels of oxygen, high altitude, increase in the exercise, and loss of lung tissue in emphysema. Red blood cells lies in narrow channels in the spleen. Red blood cell lies in the narrow channels in the spleen. And there are macrophage. We talked about the spleen being a graveyard, and then we're going to break down the globin in the hemoglobin. Heme is going to be converted into iron, and globin is going to be hydrolyzed to multiple amino acids. So the heme pigment is going to be converted to biliverdin, biliverdin converted to bilirubin. So this is a very important figure here because it shows us that in the small intestine we absorb the iron, we use it for erythropoiesis, and then the spleen is going to break down the old red blood cells. Remember when we talked about the graveyard of the red blood cell? So the graveyard of the red blood cell is as a result of damaging and breaking apart the red blood cell. So we don't just throw it away. You know, after you drink your can of soda, you don't throw it away in the garbage. You recycle it. So now we need to recycle those old, remember we said they are very old and damaged? Those old and damaged red blood cells, we don't throw them away. We recycle them. So the cell fragments are going to be going back into the body to be phagocytosed, and hemoglobin is going to be recycled. Well, you know how we're going to recycle hemoglobin? Because hemoglobin is made up of heme and globin. Heme and globin. Heme means what? Iron. So the heme part, we're going to make iron from it. The globin part is we're going to break down the globin protein into its building blocks. What are the building blocks of globin? Amino acids. We're going to recycle it, make more amino acids, and use those amino acids for our own survival. But the heme is going to be broken down into iron and biliverdin. The iron is either going to be stored, reused, or sometimes lost by the menstrual cycle, for example, or bleeding. But the biliverdin is going to be converted to bilirubin, and bilirubin is going to be added to the bile, and bile is going to be secreted in the fecal matter. That's how we get rid of the toxic bilirubin and the biliverdin. Polycythemia. We mentioned that if you have too much of any cell, it means there's cancer. Polycythemia, meaning that there is excess red blood cell. Please remember that. Whenever you hear polycythemia, that means there is an increase in the number of red blood cells. Now there is primary polycythemia and secondary polycythemia. Primary polycythemia and secondary polycythemia. Question. Primary polycythemia is known as polycythemia vera. Polycythemia vera. What does polycythemia vera mean? It means that there is a cancer in the red bone marrow that is increasing the red blood cell count as high as 11 million red blood cells. As high as 11 million red blood cells per microliter. So there's an 80% increase in the hematocrit. Hematocrit should only be how much? 45. Now it becomes how much? 80%. Secondary polycythemia means that there's a dehydration or emphysema. That's what I was talking about, emphysema or nicotine. You smoke cigarette, it prevents the red blood cell from unloading the oxygen. Red blood cells are going to choke and unable to unload their oxygen. Then the red bone marrow has no way of knowing that. The red bone marrow will only sense that there is not enough oxygen in the blood. There is not enough oxygen in the blood, so we will make more what? When the bone marrow notices that there is not enough oxygen in the blood, it will compensate by doing what? Will produce more red blood cell. So this is secondary polycythemia. Primary polycythemia is polycythemia vera when there is cancer. So the reason why red blood cell count is increasing is because of cancer cell. But secondary polycythemia is the reason why red blood cell count is increasing is because not of a cancer cell, but because of a pre-existing condition, like a lung disease, like emphysema, like smoking, okay, like dehydration. So dangers of polycythemia is going to be increased blood volume, viscosity, which leads to blood clots that will dislodge and move away. Those are known as the embolus. It's going to be blood embolism, blood embolus. Any questions? The red blood cell count normal range is 4.6 to 6.2 million, while women have a lower red blood cell count. Okay, now if you look at anemia is a drop in the number of the red blood cells, a severe drop in the number of the red blood cells. Can be due to the hypoxia, and sickle cell disease is an example of a disease where there is a structural defect in the red blood cell. There is a structural defect in the red blood cell, meaning that the red blood cell was supposed to be biconcave. Red blood cell is supposed to be biconcave and doubly depressed. Biconcave and discoid. People who have sickle cell anemia, they do not have a discoid and biconcave cell. True or false? True. And what's the reason for that? It's because there are proteins that ensure that the structure of a red blood cell is biconcave and discoid. Now, proteins, if there's a mutation, they will not come out the healthy and the proper structure that they're in. Is that clear? Now, if they have that particular type of mutation, there will be no ability to make biconcave and discoid red blood cells. And the reason is there was an amino acid known as glutamic acid. Glutamic acid. Glutamic acid is the amino acid that helps the red blood cell become biconcave and discoid. So every cell that is biconcave and discoid has which amino acid? Sorry? Glutamic acid. Glutamic acid. Now some people will have a mutation and there will be no glutamic acid, and instead of the glutamic acid they have another alternative amino acid known as valine. Valine. Yes. Yeah, so the glutamic acid is the amino acid that helps. Glutamic acid, the glutamic acid known as GLUE, that's the abbreviation for it, is the one we need to become biconcave and discoid. But if there is a mutation, and we no longer, there is a mutation, we no longer have glutamic acid. Now what will we have? Valine, also known as Val. Valine. Valine. Are we clear? That's known as valine. It differs only, we said, with valine. So now if you look at the different blood types, there are different blood types based on the antigen that is found at the surface of the red blood cell. So let's go over this. Okay, everybody write this down. If you look into every red blood cell, on its surface, a red blood cell on its surface has an antigen. Some have A antigen, some have B antigen, some have AB antigen, both. A and B, both. A and B antigens, and some are bare, naked. They have nothing on their surface. We call that O antigen. Is that clear? That is called O antigen. That is known as O antigen. Everybody get the 4 antigens? You have the A antigen. You have the A antigen and the B antigen, the AB antigen, and the O antigen. You have the A antigen, you have the B antigen, you have the AB antigen, and you have the O antigen. You need to understand, you will not have all of these red blood cells inside your body. You only will have one. So your immune system has antibodies against what it doesn't have. So if you are blood type A, you will have an antibody against A. Will you have an antibody against A? Antibody meaning you will destroy. An antibody will destroy. A antibody means it will destroy A. B antibody, it means it will destroy B. Do you get it? Somebody with blood type A. Somebody with blood type A. Do you think will they have an antibody against A? Will they have antibody against A? No. They will have antibody against who? B. So people who are type A, they will have they will not have anti-A, and they will have anti-B. Anti-B, antibodies. Anti-A, antibodies. So, if you're type A, you're never going to have anti-A antibodies, because that means you will end up killing your own self. But you will absolutely have anti-B antibodies. True or false? So if somebody and you are type A and they give you type B, what is your immune system going to do to type B blood that was given to you? It's going to do what? I'm sorry? It's going to kill it. It's going to destroy it. The antibody, if you're type A, they give you B, is going to destroy it. If you're type B, will you have anti-A antibodies? If you're type B, will you have anti-A antibodies? Yes. If you're type B, will you have anti-B antibodies? You will not. If you're type AB, will you have anti-A? You will not have anti-A because you will kill yourself. If you if you're type AB, will you have anti-B antibodies? And if you're type O, there's really nothing on your surface. Will you have anti-A antibodies? Will you have anti-A antibodies and there is nothing on your surface? Will you have anti-A? Yes, you will have anti-A antibodies. So you don't have A. You don't have A, you will have anti-A antibodies. Will you have anti-B antibodies? Excellent. Now, let's say you give somebody who's blood type A, you give them blood type B. Or somebody blood type B, you give them blood type A. Or somebody blood type O, you give them blood type A or B. What is that? Sorry? Blood type B. If you give somebody blood type A, you give them blood type B. If you are blood type A, somebody gives you blood type B. You are blood type B, somebody gives you blood type A. You are blood type O, somebody gives you blood type A or B. What will happen? Nothing? Nothing will happen. So if somebody gives you blood that is different from you, remember, you are blood type A, you're supposed to get A, but they gave you B. You're blood type B, you're supposed to get B, but they gave you blood type A. You're blood type O, you're supposed to get blood type what? If you're blood type O, you're supposed to get which blood type? You only can have yourself. You can't be blood type O and you take A and B. You only get O. If you A, you only get B. If you're blood type A, you only get A. If you're blood type B, you only get B. Now I'm asking you if they make a mistake. If you're blood type A, what do they give you? So what do you mean by harmful? Very good. So how is it harmful? What happens? The antibodies are going to damage and they're going to do something known as agglutination. Agglutination. That's known as agglutination. That is agglutination. Agglutination is when you give somebody the wrong blood type and all of the antibodies are gonna glue up all these wrong red blood cells and then damage them. This is called agglutination. So now we will see a photo of that when we go over but when it comes to blood type O, it can only receive blood type O. What's the name of this A here on the surface? The B on the surface. The A on this surface, the B on this surface. What do we name those on the surface? Those are what? Those are antigens. Those are antigens. And the Y shape that will clip them together and cause agglutination, what are they called? They're called antibodies. Students have a problem remembering the difference between an antibody and a antigen. Antibody and an antigen. Okay, so red blood cells, you have A and B, and those are you see those are the blood type O sugars, those are blood type B sugars, those are blood type A, and those are blood type A and B. So blood type A person has A antigens. Blood type B person has B antigens. Blood type AB has both A and B antigens. And blood type O has neither A or B. So that's what happens. What is this? What does this blue bottle have? What does the yellow bottle have? Well, they have the antigens, but you're not specifying which antigen do they have. The blue one has an anti-A antigen. The blue one has the blue one has anti-A antigen. The blue one has anti-A antigen. And the yellow one has anti-B antigen. The blue one has anti-A antigen and the yellow one has anti-B antigen. It's very obvious. So now, if you put anti-A, if you have, those are anti-A antibodies. The blue is anti-A what? What are anti-A what? Anti-A what? Anti-A antibodies. So if you put them in a blood that has A antigens, what are they going to do to it? What are the anti-A antibodies going to do if you put them in blood type if you blood type A? Your blood type A, you put anti-A antigens on it. What's going to happen? It's going to cause agglutination, like this. That's agglutination. So now, this is a person who is blood type A. That's a person with blood type A. That's a person with blood type A. When they put and you know you don't know, this is just to tell you, but that's how they find out what your blood type is. So let's say you have a mysterious blood sample. You don't know, is it type A, B, AB, or O? You bring the anti-A antibodies and you drop it on the blood type A. If it agglutinates, that means that blood was type A. You bring anti-B antibody. If you drop it on the blood and then the blood agglutinates, that means this blood definitely has B antigens on it. So now, if you look at blood type AB, if you put anti-A on anyone, it will damage it because it has A and B. So if you put blood type A on an AB, it will agglutinate. If you put blood type B and you put anti-B on it, it will agglutinate also. So that's how we tell. But if you put anti-A antibodies and anti-B antibodies on blood type O, it will never ever affect it. Because blood type O doesn't have A antigens and doesn't have B antigens, and there is no such thing as O antigens. There's no such thing as O antigens. Is that clear? Antibodies or agglutins, those are anti-A and anti-B antibodies. So they appear 2 to 8 months after the birth, and the maximum concentration is by 10 years of age. You see, this is the agglutination. Those are the agglutinins. Talk about the agglutination. And you see what happens if you add blood. If you look at this figure here, this is a person whose blood type what? That's the person whose blood type B. That means it has anti-A. How can you be blood type B and you have anti-B? You blood type B and you have anti-A. So they injected that person with blood type A. So now it has anti-A, and you will see that there's going to be the donor red blood cells are going to be agglutinated by the recipient's plasma. And that's going to be the agglutinated red blood cells that are going to make like a blood clot. Okay, so we talked about that type O. Okay, now this is the hemolytic disease of the newborn. Now, why do people say blood type A, blood type B, and sometimes A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative? What is positive, negative? The positive and negative refers to the rhesus factor, Rh. Now, if you have a rhesus factor as a mom, now that's the whole separate convention. That has nothing to do with blood type A or B or O. The rhesus factor is an antigen that's going to be at the surface of the blood cell. Rhesus factor is at the surface of the red blood cell. Do you get it? Rhesus factor is found at the surface of the red blood cell. If you have a rhesus factor, that makes you Rh positive. Rhesus factor means you are Rh positive. Rhesus factor means you're Rh positive. Now if you look, you will see that the rhesus factor is Rh positive, and if you have no rhesus factor, you will be called Rh negative. Is that clear? So the issue here is not the baby, it's the mom. And you know, as a baby, your blood is circulating through the blood of your mom. You don't have your own lungs yet. Your own lungs are not in exposure to air just yet. Your lungs and your heart is shuttling the blood to your mom's red blood cells and your mom's circulatory system and your mom's lung. She's going to breathe for you. So if the mom has a Rh positive and the baby has no Rh, will the mom kill the red blood cells of the baby? No, because Rh minus means you have no Rh only. So you're not going to have antibodies against nothing. And if the baby is Rh positive, and so is the mom, Rh positive, will the mom kill the baby? No, because we both have it. The problem is when the mom is Rh minus. The mom has no rhesus factor. The mom is Rh negative. If the mom is Rh negative and the baby is Rh negative, will the mom have antibodies against the Rh negative? No, because you can't have antibodies against nothing. Rh negative means it has no Rh. But if the mom is Rh negative and the baby is Rh positive, will the mom destroy the baby now?
If it's the first pregnancy.
If the first pregnancy, we're going to develop the antibodies. But on the next pregnancy, if the mom is Rh negative, that means it has antibodies against Rh. So the mom has antibodies against Rh. So the mom is going to send out antibodies from the first pregnancy. So in the first pregnancy, you're not going to die. God's going to give you a second chance. But the mom has to get the shot. The shot known as RhoGAM. If the mom gets the RhoGAM shot, she really has nothing to worry about. The least abundant form element. So those are the white blood cells. And we talked about that. The neutrophils mainly target bacteria, eosinophils. So the neutrophils are 60 to 70%, and they are barely visible. Eosinophils, these are large, rosy orange granules, bilobe. Basophils are large, abundant, violet granules. Lymphocytes are variable amounts of bluish cytoplasm, and monocytes, those are the largest white blood cells. We discussed those. There you go. Neutrophils, they are increased in bacterial infections. Neutrophils are increased in bacterial infections. Eosinophils are increased in parasitic infections, while basophils are increased in numbers in viral infections like chickenpox and sinusitis and diabetes. Then the neutrophils, the eosinophils, those are the basophils. Lymphocytes, those are increased numbers in diverse infections and in the immune response. So they destroy the cancer cell, foreign and virally infected cells. Monocytes have increased numbers in viral infections and inflammation, so they leave the bloodstream and they transform into macrophages. So there are antigen-presenting cells who take fragments of these pathogenic microbes and then presenting them for cytotoxic cells to come and kill them. Those are the agranulocytes, the lymphocytes, and you see the leukopoiesis, the same way we said erythropoiesis, means making of red blood cells. Leukopoiesis means you produce the white blood cells. So the red bone marrow stores and releases the granulocytes and the monocytes. Circulating white blood cells do not stay in the bloodstream, and we said that they mature these. So that's the photo I was talking about earlier. That's the most important figure that I want you to get today. Yeah, you can't leave the classroom for that long. Yeah, I'm sorry, you can't do that. No, you can stay, but you have to speak with me after class, please. So that's the most significant photo that you must get. You remember what I was trying to tell you earlier? When you have dough, milk, and flour, and butter, and then you make cookies, you make a pancake, you make a cake, you make anything that you make from it. But remember, you have to have the original dough. And from there, you build up whatever you want to make. So my original dough is my stem cell, my pluripotent, multipotent. What's potent mean? Potent means potential. It has multiple potential. What's the multiple potential? Potential of becoming a eosinophil, potential of becoming a basophil, potential of becoming a neutrophil, potential of becoming a monocyte, potential of becoming a B lymphocyte, potential of becoming a T lymphocyte, potential of becoming a natural killer cell. Remember how I told you when you were in high school, you all had the potential of being a high school graduate. When you go to college, now you commit to one line. You see what's happening here? We went up the eosinophil, so now you're gonna become a police officer, a medical doctor, you're gonna become a engineer, a nurse. So now you're gonna commit to one line. So the original cell, the potent stem cell, will either go in each colony forming unit. Remember I told you, the first step is called the colony forming unit. So what happens is the colony forming unit, that's the eosinophil. And so remember, if you go to the colony forming unit, can you reverse? Can you go back to becoming a stem cell after you were a colony forming unit? No, you have to go only forward. Can you cross over? Can you go from like a eosinophil to a basophil? Can you do any of that? No, you just go forward. And that's kind of the lifeline. So the eosinophil, you become a myoblast, then you become a eosinophil. Then basophil, then you end up with a basophil. Then you commit into a neutrophil or a monocyte, then you a monocyte. The lymphocytic colony stimulating factor and the colony, sometimes they call it colony stimulating unit and sometimes colony forming unit, same thing. And then we're going to become B lymphocyte, T lymphocyte, and natural killer cell. Leukopenia and leukocytosis. Leukopenia is when there is a drop. Leukopenia is a drop in the white blood cell count. So it causes radiation, poison. So if you undergo like a cancer, chemotherapy, and there's some form of cachexia and radiation or some infectious disease, you will see that your white blood cell count is gonna drop. Leukopenia means drop. Leukocytosis from site cell, so when there's an increased genesis, leukocytosis, that is high white blood cell count. Leukemia is a cancer of the white blood cell, meaning if you go above 10,000 white blood cells, that's leukocytosis. Below 5,000, that is leukopenia. Leukemia is cancer of the hematopoietic tissue that usually produce an extraordinary high number of circulating leukocytes, the precursors. Myeloid leukemia is uncontrolled granulocyte, while lymphoid leukemia is uncontrolled agranulocyte. So acute leukemia appears suddenly and progresses rapidly, and death is within a month. Chronic leukemia is undetected for a month, and the survival time is usually, unfortunately, only three years. So we talked about the white blood cell count. And lastly, we're going to end up with this very important concept of hemostasis. Hemo means blood, stasis means stagnant, constant. Stasis means constant. And in order for you to preserve your blood, you need to have the mechanisms to clot this blood. Otherwise, you will bleed endlessly and you will lose all the red blood cells and you will not survive, and death will become imminent. So there are built-in survival, first aid systems to help you until the ambulance gets there and they give you a supply of blood, but long before that, your body has that adaptive mechanism. Number one, vascular spasm. That's very important because if the blood vessel is not squeezed, it's going to lose blood endlessly. So we need to perform vascular constriction and spasm. Platelet plug formation, and that's the thrombocytes coming to perform the band-aid. And blood clotting, that is the coagulation and red blood cells piling up. Platelets play an important role in all three: vascular, platelet plug formation, and blood clotting. So platelets are small fragments of megakaryocytes, and the normal platelet count is usually from 130,000 to 400,000. And these are platelets, we talked about platelet plug formation. And obviously, after we perform the blood clotting and then we healed, there's no need to perform the platelet plug. We need to dissolve the platelet plug. We need the platelet plug dissolution. So we'll talk about the streptokinases and heparin that will loosen up the blood. So thrombopoiesis, the same way we had leukopoiesis, white blood cell genesis, red blood cell genesis. Now we have platelet, red blood cells, platelet making. So that develop receptor from the thrombopoietin. Vascular spasm, we talked about it, that's the prompt constriction of a broken vessel. And the cause for that is the pain that is stimulated with a neuropathic pain or a nociceptive pain. And those are, you need to know the triangle. That's the triangle of hemostasis. Whenever you're bleeding, three things will happen. Number one, vascular spasm. Number two, platelet plug formation. The band-aids will apply. And lastly, there will be thickening, and the blood will become like ketchup, really thick, so that it becomes harder for it to move. So people who are sedentary, they don't exercise, they don't run a lot, what happens is their blood is going to loosen. It's going to become really thick. They're always going to get blood clots. Platelet plug formation, that is when there is a broken vessel, exposes the collagen. Okay, in coagulation or clotting, so that's the last and most effective defense, and that is to ensure that if, in case we have lost blood and the platelets did not prevent blood loss, then we ensure that the blood that we have is moving slower than it usually is to prevent its loss and to prevent that it's not being saved. There are two pathways, an extrinsic and an intrinsic pathway, but they both converge at the same step, which is activating factor number 10. You really need to know that. They always ask about that, and you will see it in your homework. I told you, it's very clear how to prepare for the exam. All these homework questions, you really master them really well. And the question will always ask whether your extrinsic mechanism, meaning that the injury was from the outside. Intrinsic is like an external hemorrhage. Intrinsic is like an internal hemorrhage. Well, whether extrinsic or intrinsic, you have to convert at a factor known as factor X. And here you will see that in the end, the goal is to factor X is going to convert prothrombin into thrombin. And thrombin is going to convert fibrinogen into fibrin. And fibrin is the one that's going to make the fibrin mesh work. Remember we said serum? Serum is different from plasma in the sense that both are identical. Serum is only missing one protein. What is it? It's missing only one protein. It's known as fibrinogen. So we're doing the extrinsic factor to activate factor 10. Factor 10 is going to convert prothrombin into thrombin, and thrombin is going to convert fibrinogen serum into fibrin. Very good. Calcium is required for both pathways. They always ask about that. Calcium is required for the extrinsic and intrinsic mechanism. Rapid clotting. Each activated cofactor activates many more molecules in the next step. So we talked about that. X. Okay, so now the positive feedback. What does a positive feedback mean? It means that if one platelet sticks, the second one on top of it, the third one on top of it, the fourth one on top of it, that's positive feedback. Red blood cell. Thrombin is going to make fibrinogen cause become fibrin. Then there's a fibrin meshwork. So one red blood cell, two red blood cell, three red blood cell, and they start to attach severely. Platelet-derived growth factor secreted by platelets and endothelial cells. Fibrinolysis is the dissolution of a clot. Factor 12 speeds up formation of kallikrein enzyme, and kallikrein converts plasminogen into plasmin. Plasmin is what's going to soften up the clot that has been formed. Because we form clots temporarily to prevent blood loss. But we don't want clots remaining in our blood. We're going to get a blood clot now. We're going to get a heart attack. So we need to dissolve it with plasma. So there you go. You see this is also one of your figures for the lab practice. You see that kallikrein is going to convert plasminogen into plasmin. And plasmin is going to dissolve the fibrin polymer into this degradable mesh. Okay. Heparin is secreted from basophils and mast cells and interfere with the formation of prothrombin activator. But now we want to dissolve and we want to repel the platelets from binding. Now there are clotting diseases that we must all be familiar with. And that's the hemophilia. Everybody needs to know about hemophilia, and that's somebody who bleeds to death endlessly. That's a deficiency of any clotting factor. It can shut down the coagulation cascade. So the hemophilia is a family of hereditary diseases characterized by deficiencies of one factor or another. Sex-linked recessive diseases is a sex-linked recessive disease that is found only on the X chromosome. So if you are a man, you're in trouble because men only have one X chromosome. Men have one X and one Y chromosome. So if the X chromosome is linked to the disease, you will have hemophilia. But women are lucky because they have two X. If one X has hemophilia, the other one is going to be healthy and she will be heterozygous carrier. But men, only one. It takes only one X chromosome that is suffering from hemophilia, and you will see that you will suffer from the disease. So hemophilia, that means you're missing hemophilia A, you're missing factor 8, that's 83% of the cases, and hemophilia B is missing factor 9. Hemophilia C is when you're missing factor 11. But at the end of the day, you're having a problem in blood clotting. A hematoma is a blood tumor, and that happens when you fracture a bone. So when you fracture a bone, the bone is surrounded with blood vessels and nerves. So you break the bone, you break the nerve, you break the blood vessel, the blood starts spatter all around the injury site, and you have a big liquid, big tumor, big blood clot. So this hematoma is an indication of clotted blood in the tissue, particularly in the bone tissue. Thrombosis is abnormal clotting of an abnormal blood vessel. And sometimes deep vein thrombosis is when a pregnant woman, because you know a pregnant woman's blood is much hypercoagulable than if you're not pregnant. Why? Because your blood is too thick because you're breathing for yourself and for the baby. So every mom who's pregnant needs to get a shot of heparin to loosen up the blood so that she doesn't get a heart attack and die like my cousin who passed away because there was no heparin. This is in my country, not here. Because there's no heparin. We looked all over the pharmacies, all over the hospitals. Black market is not there, so she just died. And when I did my gastric sleeve, I was very big. When I did my gastric sleeve, they gave me so much enoxaparin shots. I said to my mom, look, she needed just one of those. They gave me like 50 of those. You know those little tiny enoxaparin shots that you have to give yourself? I said to my mom, she needed one of those. Yeah. And this deep vein thrombosis is going to lodge. It comes from the deep vein and it's going to travel and it's going to clog what? It's going to clog the pulmonary artery.
Professor?
Yeah.
What country are you from?
Sudan. Yeah, it's not good there. It's bad. And it's bad and it's getting worse. That's the sad thing. It's not getting better. It's one thing if you're bad and stable, it's okay. But if you're getting worse, it's not good. It's really bad. And war, and rape, and killing, and murder, and it's very bad. It's very bad. I'm very sad. Okay, embolus is anything that can travel in the blood and block blood vessels. Infarction is tissue death. It may occur if the clot blocks the blood vessel, so the blood cannot now pass into the tissue. So the tissue will suffocate because it's waiting for the blood. Okay. Blood clotting is to prevent formation of clots or dissolve existing clots and preventing clots. We take vitamin K is required for the formation of the clotting factor. And aspirin suppresses the thromboxane. So that's how we end up loosening the blood. And lastly, dissolving the clots that have already been formed. We use streptokinase. It's an enzyme that's made by the streptococcus bacteria used to dissolve the clot in the coronary arteries. Tissue plasminogen activator works faster, it's more specific, and now is made by the transgenic bacteria. This is like a pharmacological advancement. We make the bacteria, this is transgenic DNA technology, and we make the bacteria make for us antibodies and make for us the streptokinases. Hementin is produced by some leech, but we don't use that much. We use the streptokinase. Any question? Any question? Okay, so let me take the attendance before.